Hello students, how are you? I hope you all are fine. So welcome to my next lecture, grade 12, chapter 5. Name of the chapter is Principles of Inheritance and Variations. So today we will discuss next topic of the chapter. Let's start with monohybrid cross. I told you in the monohybrid cross only one character will occur in the cross. In the given figure you can see tall plant is there and dwarf plant is there. Tall and dwarf, these both are the homozygous condition. Capital T, small t. But capital T is present in dominant condition and small t is present in the recessive condition. After the mating in the F1 generation, then heterozygous gametes will produce. Heterozygous, that means capital T and small t. But these all are tall. Why? Because tall is dominant to the dwarf. Okay. And after the F2 generation, you can see phenotypic ratio, that is having 3:1. Because capital T is showing dominancy to the small t. That's why 3 are the tall and 1 is dwarf. And in the genotypic ratio, you can see 1:2:1. Now, production of gametes and formation of the zygotes, that can be done by Punnett square. Punnett square is developed by British scientist R.C. Punnett. Punnett shows graphical representation by calculating probability of possible genotype in a genetic cross, and the gametes are present on the two sides, top row and left column. In the given figure you can see. Now in the F1 generation you will see 50% gametes will produced. In F2 generation, 3/4 are tall, 1/4 are dwarf by phenotypically. And 1/4 raised to half raised to 1/4 ratio, that is genotype, between capital T, small t, and small t. Now test cross, that is the cross between the hybrid and its homozygous recessive parent, that is called test cross. It is usually to identify the genotype of the hybrid. Now, Mendelian's law of the heredity. These rules were proposed by Mendel. There are two rules, principles or law of the inheritance, in which first law, that is law of dominance, and second law of the segregation. So let's start with law of dominance. First, in which characters are controlled by a discrete unit. That unit is known as the factor. And we know these factors are present in a pair. Okay? If these factors are in dissimilar pair, then one member of a pair, that is dominant, or other is the recessive. Dominant dominate over the other. And used to explain the expression of only one of the parental character in the monohybrid cross and expression of the both in F2 generation. Also explain proportion 3:1 in the F2. Now law of the segregation. It states that when a pair of factor for a character brought together in a hybrid, then they will segregate during the formation of the gametes. Okay, and alleles that do not blend and both characters are recovered in the F2 generation, and in F1 generation only one will recover. Okay, factor which is present in the parents that segregates, and gametes receive only one of the two factors. Okay, either tall or either dwarf. In the homozygous parent, one kinds of gametes are there, but in the heterozygous parent, two kinds of gametes, each one have one alleles with equal proportion. Okay. After that, incomplete dominance. As the name indicates, incomplete. That is not complete, partially. Correns discovered incomplete dominance in the Mirabilis jalapa. It is also known as the partial dominance or semi-dominance. The inheritance in which alleles for a specific character is not completely dominant over the other alleles. Okay, that is called as incomplete dominance. Example, snapdragon and Arianum species in which crossed occur between the true breed of red flowers and the white flower. Red flower capital R, white flower small r, small r. And in the F1 generations, pink color flower will produce, that is capital R, small r. And after self-pollination in F2 generations, you will see 1 raised to 2 raised to 1. That means red raised to pink raised to white. In the pink, you can see incomplete dominance is there. Neither red, neither white. And genotype ratio same as the Mendelian ratio and phenotypic ratio different than the Mendelian ratio. Because in the Mendel's law, there was 3 raised to 1 and in which 1 raised to 2 raised to 1. Incomplete dominance can be seen in snapdragon. In the given figure, you can see parents are there, which consists capital R, small r, and after that gametes will produce, which consists capital R and small r from the white. And after the mating in the F1 generation, pink flower will produce, which is having red as well as white character. Okay, that is showing the incomplete dominance. Okay, and after the self pollinations, that gamete will produce red, 1, 2 pink and 1 white. Okay, in this picture you can see the phenotypic ratio is 1 raised to 2 raised to 1 and the genotypic ratio is 1 raised to 2 raised to 1. Both are same. Now codominance. Both the alleles for a character are dominant and expresses its full character. That is codominance. You can see codominance in AB blood group of human being. Blood group in human being are controlled by 3 alleles of a gene I. They are IA, IB and small I. The ABO locus is located on the chromosome 9 and IA is responsible for the antigen A and IB responsible for production of antigen B and small I that does not produce any antigen. IA, IB are codominant and dominant over the small I. That means small I is a recessive part. In the given table you can see if the blood group is A, then genotype will be IA or IA small I. If the blood group is B, then genotype is IB or IB or small I. In AB blood group, genotypes are IB. And in the O blood group, you can see the genotypes are small I. ABO blood grouping which show multiple allelism. 3 alleles govern same character. Okay, the multiple alleles is found when populations studies are made. In the multiple alleles, single gene product may produce more than one effect. For example, starch synthesis in the pea seeds that is controlled by a gene which is having 2 alleles, capital B and small b. And starch synthesis is effective if homozygous capital B which produce large starch grain and homozygous small b which having lesser efficiency in the starch synthesis and seeds are wrinkled because that is in the recessive condition. In the heterozygous, then it's capital B and small b, then you can see round seed and intermediate size. Okay, so rest part we will discuss in the next lecture. Till then, stay at home, stay safe, be happy. Thank you.